See, from 2023 or so, this chemistry in everyday life has been added in JEE advanced syllabus. Okay, before that it was in main syllabus only. Now they have removed from mains, but is now very much part of your advanced syllabus. And ever since it was added, every year almost we are having one question. See, J23, 24. Okay, now we will try to solve it. Again in 24, there were two questions on the similar, on the same chapter. Okay, with a combination of other chapters, of course, but yes, they were asked. And if I go for mains, I have attached these in today's discussion. In the end, you will come across with all these, some of the questions जो main से पूछे गए हैं. Okay. In backside, you will find these all. Besides this, you have to compulsorily complete the exemplar of chemistry in everyday life. This, I hope all of you are able to see the screen. So whatever material you will get from your centers, that is very good, wonderful. You can solve from module, again, very nice. But besides that, compulsorily solve this NCERT exemplar, all questions, including the short answers, matching types, and the reason assertions, long answers. So you can see you have a very authentic collection of questions for this particular topic from NCERT itself, 101 questions. Okay, and plus they are from NCERT, so they are very authentic, that around what concept we can expect the questions. Okay, so read NCERT at least 2 to 3 times. I am reading it with you and will explain everything. First of all, I will request you all not to take this particular chapter lightly. If anyone has new joined the meeting currently, please take your NCERT in your hand. We will read and we will mark important points and will explain wherever required. Okay, don't take this chapter lightly, because if it is there, you can score it directly by sheer 2 to 3 times reading. Overall, by the time you can score the question. Yes, I think it was Ashish or Paul. Someone just rose the hand. Who was it? Was it Paul or was it? Yes, Paul. What is the problem, beta? Is there anything you want to say? Nothing? Okay, fine. So then let's start. Anyone join later, get your NCERT. So I don't see anyone having any unmute and answer, questioning anything. So I'll start the reading. It will take some time. So, like chemistry in everyday life, I just said that don't take it lightly, because it is a chapter which could score in exam. But besides that score, it is again a topic which will give you an actual application of chemistry that you study entirely in your day-to-day life. Okay, it is not something which should be ignored. And the purpose and objective, well and good. Let us start. From living perception to abstract thought, and from this to practice. Take this is something chemistry in everyday life coming here. By now you have, I'll read every line as far as possible. Wherever I think it is not that much required, I'll ask you to read and we'll wait. So by now you have learned the basic principle of chemistry, right? The basic principles of chemistry, and also realize that it influences every sphere of human life. The principles of chemistry have been used for the benefit of mankind. Think of cleanliness. The materials like soap, detergent, household, bleaches, toothpaste, etc. will come to your mind. Now, what actually happens is if I'll ask you where you see physics, you will give all around the space, all everywhere I watch, I can find a principle of physics applicable there. If I'll say what is the use of maths, you will say day to day life, everywhere there are calculations involved, there is a basic understanding required which maths is giving us. Very good. When I ask where is chemistry, most of you will say, oh, that is an abstract type of thing, we just do it for the sake of syllabus and nothing that is so very interesting or fascinating for us. Right now, that is a very very bad understanding of your subject, because if you see, initially whether it was physics, right, or it was, say, chemistry, or it was biology, they were all together, but for the purpose of simplifying the overall science so that we can get into the depth, like one person cannot get into the depth of all three at the same time, right? Physics, chemistry, biology, one person cannot have the depth of all three. So somewhere the division was not intended to make us fight and ignore one or the other branch, whether it is biology or chemistry or physics. See, if you see biology, when you study, you know these are the problems. For example, let me talk about specs, right? When someone is there and he or she has some eyesight issues, that is told by biology. Physics chapter optics gives the solution. Bole, if you could have a lens of this power or that power or this type, you can deal with this biological condition or a problem of a particular eyesight. When this is giving a solution, this is giving a problem, chemistry is somehow bridging the gap. If you see what power to be used in optics, what type of lenses will be required, physics is told chemist. Now chemist prepared the polymer which is like best suited for those glasses, and it gives you a range of polymer which could be used for these specs based on the requirement of the patient or the person, plus the cost he or she wants to give. So we are practically acting as a junction between different sciences. It is not just that. If you see chemistry, where is chemistry in everyday life? Then medicines, wonderful. Like all of you are aware of chiral center. From today, make it a habit. Whenever you come across any medicine, whenever a medicine is wrote to you that, okay, take this medicine for this particular problem, go to Google, check its structure, find how many number of chiral centers are there, what are the functional groups present in them, how is it working, what are the side effects, because entire thing is based on your organic chemistry. Okay, so these are the actual examples, and these are not limited here itself. Your moods, how you feel, how you interact, how you behave under certain conditions, that feeling of, anxiety, that feeling of tiredness, that feeling of depression, that feeling of, sleepiness or overexcitement at times, hilarious, whatever you name, those feelings are all somewhere down the line chemicals functioning in your body. Of course, I will never deny along with the biology. See, if you see, you cannot draw a clear line between any type of science. It is just, if someone says no, that I am a physicist, or I'm a biologist, or I'm a chemist, they certainly, if they are very knowledgeable person of their subject, they will always understand there is no clear demarcation between these branches, and all of us have to work together in the sync so as to work for the betterment of humanity rather than fighting that which stream is better. They are all required. The only thing is, as and when we start studying, our entire focus ultimately lies only on plus 3 minus 1 or the partial marking or the integer. We usually don't think beyond that fact, and there we lose interest in all these topics. Otherwise, I'll say this is one of the most important topic which a 12th student should know in depth, because after 10 or 15 years, this is the topic which will actually carry, or which you will carry forward with you in your day-to-day life. Okay, all those chemistry things that you're studying now, aldehyde and ketone and carboxylic acid and whatnot, they are all ultimately applicable in your day-to-day life through this chapter. So please don't ignore this chapter. The attendance itself is showing that how ignorant people are towards this chapter. Okay, don't do that. If it is part of your chapter, you should study. Now, they are also trying to make the same impact that if you open your eyes from morning and you put your leg into slippers, slippers are polymer chemistry. If you are wearing specs, then those are the polymer chemistry. You move to washrooms, you see every possible thing in the washroom, from ceramic tiles to, say, taps, right? And how they're functioning, to the glass with which the lights or the bulbs are made. Okay, toothpaste, or you see the bristles of those toothbrushes, or what type of handle they are making, the tongue cleaner. Oh, what to say? Everywhere it is chemistry. The thing is, you have to open your eyes and see it. So that is what they are saying, that it is all around. Then after that, you go to take a breakfast. Food materials, again, everything is chemistry. You are having a carbohydrate, you are having a fat, you are having protein. All are ultimately to be understood and applied by the logic of chemistry. Okay, which you have is coming back to this food materials, again a number of chemicals about which you have learned in the previous unit. The previous unit is biomolecules, will appear in your mind. Of course, sickness and disease remind us of medicines, which are again chemicals. Explosives, fuels, rocket propellants, building, and electronic material, etc., are all chemicals. Chemistry has influenced our life so much that we do not even realize that we come across chemicals at every moment. It is something which is very easily around you. You stop appreciating that. That is how the chemistry is being treated, that we ourselves are beautiful chemical creations and all our activities are controlled by chemicals. In this unit, we shall learn the application of chemistry in three important and interesting areas. This you should remember. This here, they are starting the syllabus. What are the three things that we have to learn here? Medicines, food materials, and cleansing agents, matlab detergents and soaps. Three things we will study. Now, if you see 16.1, right? 16 is a chapter. Sub point 1, they are starting with drugs and the classification of drugs. First, I'll just lay an outline, then I'll enter into each and everything. Okay, so here they are starting drugs. The entire chapter, how to break down the chapter is to be break down into, first I have to study what is drugs. Then they will talk of classification of drugs. Classification, there are three types that we will study. Then they say what is the drug and target interaction. Okay, inside the drugs, they are saying what is the drug and the target interaction. Interactions they will teach us. Drug as receptors at drug targets. Okay, functioning of basically drugs. Then what are the different types of drugs, uses, therapeutic action of different classes of drugs. Okay, so drugs only they are teaching you everything till where? 16.3.4. we are still in that analgesic, okay, antibiotics, all type of drugs we are being taught, anti-fertility drugs, right? Now we come to 16.4 chemicals in food, okay, 16.4. Now you may see 16.3, 16.2 and 16.1, they are all basically subtopics related to drugs. They are all related to drugs. Then at 16.4, we will have a deviation to another topic called chemicals in food, which is relatively small subtopic. 16.5 is a cleansing agent, soaps and detergents that we'll study. Okay. This was from an old NCRT, which they have now removed, but I want you to know and then study this. So I have added this from very old NCRT, 2004 or 5. They sometime ask this question. Okay. So before these were their fungicides and all those uh, chemicals which causes industrial accidents. Then the past JE advanced and JE mains questions are there. Okay. So coming back to where we were, starting with drugs. So the main part of the chapter is basically drugs. Drugs will constitute the main part that we have to study and understand as much as possible. Right. So drugs are chemicals of low molecular mass. Okay. So they are the chemicals of low molecular mass, 100 to 500 units. These interact with macromolecules, macromolecular target in a large, and produce a biological response. When the biological response is therapeutic and useful, these chemicals are called medicines. So they are called medicines when they are having a positive impact. Okay, when they are having a positive impact, then they are called medicines and are used in diagnosis, prevention, and treatment of diseases. Most of the drugs used as medicines are potential poisons also, if taken in doses higher than those recommended. Use of chemicals for therapeutic effect is known as chemotherapy. If we are using chemicals for some therapy, then it is called chemotherapy. So what we learned, drugs are chemicals with small molecular mass. They are basically chemicals. They attack or they interact with some macromolecular targets, large molecules, right? Large molecular targets and generate some biological responses. If the biological response are in favor of health or are for some good purpose, then they are called medicines. They can be poisonous if used beyond the prescribed doses. Then they can act poisonous. They can even be fatal. एकदम जान ले लें ये भी हो सकता है. Okay, that is a possibility. These interact with, most of the drugs are medicine usually, but can be fatal. If they are used for therapy, then the process is called chemotherapy. Use of chemicals, or this is called chemotherapy. Okay. Coming to this, here on the top, you see I have added something from Internet General. What is pharmacology? Pharmacology is not synonymous with pharmacy. Pharmacy shop is different and pharmacology is different. And the two terms are frequently confused. Pharmacology is a biomedical science. Okay, it is a biomedical science, deals with the research, discovery, and characterization of chemicals which show biological effects, and the elucidation of cellular and organismal function in relation to these chemicals. In contrast, pharmacy, a health service profession, that is medicinal shop type thing, is concerned with the application of principle. So pharmacology is a like a branch of science in which people do research and then find what chemical is in favor, how to impact it or how to use it. That is pharmacology. And then that knowledge, when applied like in a commercial way, in a profession, then it is called a pharmacy. Is concerned with the application of the principle learned from pharmacology in its clinical settings, whether it be in a dispensing or clinical career role. Okay, in clinical care role. In either field, the primary contrast between the two is their distinction between direct patient care, pharmacy practice, and the science oriented research field driven by pharmacology. The word pharmacology is derived from Greek, okay, where the pharmakon, meaning drug or poison, together with another Greek word, logia, and the meaning of study of, knowledge of. So, knowledge of drugs is pharmacology. Pharmakon is related to pharmakos, the ritualistic sacrifice or exile of a human scapegoat or victim in ancient Greek relation. Okay, so that is how this word came. Here we are going to use this word. So I introduced this, what in literature this word is. Let us start classification of drugs. Now see, the classification of drugs is done into four categories. Okay, so classification of drugs they have done in four categories. First, on the basis of pharmacological effect. Pharmacological effect matlab how the body will react to that chemical. Okay, this is the first. First classification is based on a pharmacological effect. This classification is based on pharmacological effect of the drugs. It is useful for doctors because it provides them the whole range of drugs available for the treatment of a particular type of problem. For example, if I talk of analgesics have pain killing effect, antiseptics kill or arrest the growth of microorganisms, right? So first classification is the pharmacology. They are generating an entire classification based on what type of impact this particular type of chemical have on our body. After eating painkillers, if you are having some pain, you will find that, okay, pain is gone. So it is a painkiller. Antiseptic, that some skin eczema or something is happening, then you can put some ointment. Those are antiseptics. Second is on the basis of drug action. This was how our body responds. Okay. It is based on the action of drug on a particular biochemical process. Now, fixed process for how it is having an impact. Okay. Basis of drug action. All antihistamines inhibit the action of the compound histamine, which causes inflammation in the body. Inflammation is some sort of swelling. All antihistamines, all. So it is a class of compound which inhibit histamines. Okay, action of histamine, thereby swelling inside the body, on the body, wherever you find is stopped or is reduced. There are various ways in which action of histamines can be blocked. That we'll learn later. On the basis of chemical structures, like on the basis of functional group types, it is based on the chemical structure of the drug. Drugs classified in this way share common structural features and often have similar pharmacological activity. For example, sulfonamides, see sulfonic acid amides, have common structural feature of this. Okay, this, like this. On the basis of molecular targets, that where they will impact, what type of places will they impact, right? Just say, for example, we just saw that something is attacking histamine. If then drugs usually interact with biomolecules such as carbohydrate, lipid, protein, nucleic acid. These are called target molecules or drug targets. Drugs possessing some common structural features may have the same mechanism of action on targets, and the classification based on the molecular targets is the most useful classification for medicinal science. So they may ask which is the most useful. Okay, so let us underline the classification based on molecular targets is the most useful classification that could be for science or for the doctors, I can say. So let us revise. There are four classification. First is a pharmacological effect of the drug. Second, drug action. Third is structure or the functional groups. And fourth is the molecular target, where it will attack. Okay, now moving to second subtopic, drug and target interaction. Okay, it has some impact. How it will have that impact? Macromolecules of biological origin perform various functions in the body. For example, proteins which perform the role of biological catalyst in the body are called enzymes. Those which are crucial to communication system in the body are called receptors. Carrier protein carry polar molecules across the cell membrane. Nucleic acids have coded genetic information for the cell. Lipids and carbohydrates are structural part of the cell membrane. Cell membrane matlab, if there is a cell, then this outer thing is called cell membrane. Okay. Inside there is some coded thing present like DNA, RNA thing. That is nucleic acid. Here it is cell membrane. Now cell membrane has lipids and carbohydrates. DNA, RNA has nucleic acids. Now the carrier proteins carry polar molecule across the cell. If there is some polar molecule I have to carry from cell membrane to here or from here to cell membrane, then the role is given to polar proteins, polar molecules. Okay, carrier proteins. So here enzyme and receptor are too important. Those which are crucial to communication system in the body are called receptors because a message may be sent, but how they will be received and enacted will be carried out by the receptors. Enzymes as drug targets. Catalytic action of enzyme. For understanding the interaction between a drug and an enzyme, it is important to know how do enzymes catalyze the reaction. In their catalytic activity, enzymes perform two major functions. The first function of an enzyme is to hold the substrate for a chemical reaction. Active sites of enzymes hold the substrate molecule in a suitable position so that it can be attacked by the reagent effectively. This is explained here. Okay, how it is functioning. Like, for example, this is enzyme. This is its active site. Now, substrate, we want substrate to react, but to keep it at one place, to hold it, and thereby other reactant which is coming can act upon it. For that, see, this is its active site. It is pretty much like lock and key. Okay, if we have a lock and we want to use some sort of key, right, to open it, then there will be certain grooves here. Even if you put a mirror image of this key, it will not open this lock, right? If you want to open this, it has to be perfectly adjusting into whatever type of opening is given in this lock. Similar is the situation. This enzyme has a active site or has that socket type thing which is very specific to a particular substrate. Not any substrate can come and fit in. It has to be particular substrates. Substrates bind to the active site of the enzyme through a variety of interactions such as ionic bonding. Again a question. There is a question here, right? What type of bondings are used? Substrate bind to active sites. This come here through a variety of interactions such as ionic bonding, hydrogen bonding, van der Waal, or dipole-dipole interactions. So there could be a question here. Is that clear? That substrate and enzyme bind by van der Waal, hydrogen bonding, ionic bonding, and dipole-dipole interactions. The second function of an enzyme is to provide functional groups that will attack the substrate and carry out the chemical reaction. They can give the functional group. Drug enzyme interaction. Drugs inhibit any of the above mentioned activities of enzyme. Any of the matlab, either they will somehow hinder this active site functioning, or they can somehow stop this functional group production thing. By any of the method, it will stop the functioning of this enzyme. Okay, drugs inhibit any of the above mentioned activities of enzymes. These can block the binding site of the enzyme and prevent the binding of substrate, or can inhibit the catalytic activity of enzymes. Such drugs are called enzyme inhibitors. So how they are inhibiting? They are inhibiting by blocking. Okay, they are inhibiting by blocking the active site of the molecule, because active site per he enzymes are coming and binding and prevent the binding of substrate, or can inhibit the catalytic activity of enzyme. Drugs inhibit the attachment of substrate on active site of enzyme in two different ways. Now this is important, right? How is this important? What we expect, I'm telling you. See, definitions will come. Drugs compete with the natural substrate for their attachment on the active site of enzyme. Such drugs are called competitive inhibitors. Okay, so what happens, as I said, they have a fixed site on, and in this only the substrate could go and enter. Now the drug that we want to use will have a similar arrangement. एक तरह से ये cheating कर रहा है. This is drug is doing a cheating or a धोखा. An enzyme is confused that it is substrate, but it is not the substrate. This drug goes and block the active site. So it is indirectly, I can say it is competing with substrate for the same site, that I will go, I will go, I will go, I will go. So it is called competitive inhibitor. It is inhibiting the function of enzyme. Second, some drugs do not bind to the enzyme active site. These bind to a different site of enzyme. Okay, they do not site at the actual site. Competitive inhibitors directly block the active site, but some do not block the active site. They get attached somewhere else. And there, when they attach, then the entire shape of that active site changes. Okay, so some drugs do not bind to the enzyme's active site. These bind to a different site of the enzyme, which is called allosteric site. What is it called? Allosteric site. Okay. Now these allosteric site, that they are not connecting to the active but somewhere else. This binding of inhibitor at allosteric site changes the shape of the active site in such a way that now the substrate cannot recognize. See, substrate had a memory or had an understanding that this is how the place will be and I have to enter in. But what has this inhibitor has done? It has gone here. You can imagine it to be a soft jelly-like thing and pumped. It contacted here, it entered here. As a result, this active site is now changed its shape. When change shape, now this substrate cannot enter here. So it has ultimately did similar function that, okay, substrate and enzyme should not come into contact, but the method is different. Okay, so some enzyme, these bind to a different site called allosteric site. This binding of inhibitor at allosteric site changes the shape of the active site in such a way that substrate cannot recognize it. If the bond formed between an enzyme and an inhibitor is a strong covalent bond, if it is a strong covalent bond and cannot be broken easily, okay, now which are the two bonds? Of course, both, whether it is going to allosteric site or it is going directly as a competitive inhibitor, cannot be broken, then the enzyme is blocked permanently. Okay, the body then degrades the enzyme inhibitor complex and synthesize the new enzyme. So there is an extra load on body. That entire enzyme is now lost by the body. It cannot be used any further. Okay? This happens only when there is a strong bond between the inhibitor, or sorry, the drug and the enzyme. Coming to next point, receptors as drug targets. What we were doing first, we were doing very 16.1.1, 16.2.1, enzyme as drug targets, how they are working as a like enzymes are acted upon by drugs. Okay. Now receptors at drug targets. Now drugs are attacking on the receptors, not on the enzyme. Now first we have to understand what are the receptors. So receptors are proteins that are crucial to body's communication process. It is very much like the receptors or the antennas once upon a time when the radios were there, or currently for a Wi-Fi you can say yes, there are receptors. Okay. So receptors are the proteins that are crucial to body's communication process. Majority of these are embedded in cell membrane. As I told you, this boundary, this wall of the cell is called cell membrane. So they are embedded in this membrane. See, like this. Okay, these cell membranes, they have enlarged it for us. Outer surface of cell membrane, which is in contact with the outside world, inner, which is here inside of the cell, and here is our receptor, the signal taking thing. Majority of these are embedded in cell membrane. Receptor proteins are embedded in the cell membrane in such a way that their small part possessing active sites project out of the surface of the membrane and opens on the outside region of the cell membrane. So like it is very much like you have a big किला को इंग्लिश में क्या कहते हैं, castle. You have a big castle and you have a big wall around that castle. In those castles there were small windows now through which the soldiers used to keep a watch on what is there outsider, can we be attacked or will we be attacked, that type of thing. That these are the receptors. Okay, small openings. Now these openings basically communicate how those windows used to communicate that okay, we are being attacked, come back and all those things. Same way these are the signal taking places. Okay, and interior mein they pass on that message. In the body, message between two neurons and that between neuron to muscle is communicated through certain chemicals. Between two neurons, those are the nerve cells, and that between nerve cell to a muscle is communicated through certain chemicals. And these chemicals are called chemical messengers. Okay, are received at the binding site here of receptor proteins. To accommodate a messenger, shape of the receptor site changes. This brings about the transfer of message into the cell. How its shape will change will decide what message is being given to that particular cell, how to act. Okay, this shape change decides. See, it was like binding site were like this, then it changed to this, and then when again it left, it is back to this. And when they change, then they see signals are gone inside the cell. Okay, these signals are gone. So these chemicals known as chemical messengers are received at the binding sites of receptor proteins. To accommodate a messenger, shape of the receptor site changes. This brings about the transfer of message into the cell. Thus, chemical messenger give message to the cell without entering the cell. See, it has not that it has gone inside. यहीं पर उसने message दे दिया. ठीक? There are a large number of different receptors in the body that interact with different chemical messengers. These receptors show selectivity for one chemical messenger over the other because their binding sites have different shape, structure, and amino acid composition. Again, a question. Here is a question that how they show that specificity. Receptors show selectivity for one chemical messenger over the other. If there are more than one messages coming, they might not interact all. Like there will be different receptors for different chemicals. That is what will be there. Okay, because of their binding sites have different shapes, structure, and amino acid compositions. Okay, So it is quite heavy. You have to be very focused. धीरे धीरे आप पढ़ेंगे तभी धीरे धीरे याद रहेगा. चलो, moving further. Drugs that bind. Let me enlarge. Drugs that bind to the receptor site and inhibits its natural function are called antagonist. Matlab, some chemical is coming and it will say cell to perform in a certain way, whether it is good for body, bad for body, maybe whatever. But if we do not want that type of interaction or that type of behavior from that cell, we want cell do not receptors do not convey this message to the cell and stop that working. For that, we generate certain drugs, they bind to the receptor sites, and now that receptor site is blocked and inhibit its natural function. These are called antagonists. These are useful when blocking of message is required. There are other type of drugs that mimic the natural messenger by switching to on the receptor. Now see, these antagonists will be used when we want to stop, when we want to stop the process. For example, if I say that thyroid is generating too much of hormone, not a thyroid itself is a hormone. Pancreas are generating too much of certain hormone. We do not want that many hormones to be created. We want to stop. So we will send these antagonists. They will stop or inhibit the natural functioning of those receptors, and those certain chemical which was generating as hormone overflow ho raha tha, it is stopped. Okay, so the message is blocked. Other way, if someone is having lack of certain hormone, for example, diabetes, we want insulin to be produced more. Okay, now the message to those generators is not given properly, and as a result, the amount of insulin produced is not sufficient for keeping our health in a proper way. Now we want that, okay, please create more insulin, please create more insulin. So here we introduce certain chemicals which will mimic, which will enact as the natural messengers by switching on the receptor sites. They will act as what should I say, as those actual messengers. Drugs that mimic the natural messenger by switching on the receptor, these are called agonist. So there are two types. One is ड्रग्स में हमने यहाँ दो नाम पढ़े हैं. ड्रग्स में दो नाम पढ़े हमने, एक है एंटागोनिस्ट और एक वर्ड हम पढ़ रहे हैं एगोनिस्ट. एगोनिस्ट increases the functioning, antagonist decreases the functioning. Therapeutic action of different drugs, how they will do that positive thing on our body. In this section, we shall discuss the therapeutic action of few important classes. First is antacid. Commercially, it is like, what should I say, wealth. Antacids are like, it has made people billionaires and billionaires over a period of time. Overproduction of acid in the stomach causes irritation and pain. Now what happens that our stomach, if I could just draw, is somewhat like this. Okay. Now this stomach has a very thick inner muscular lining. And here we have acidic pH. Inside here is acid. But if this acid's production is increased, okay, why it is increased, how it is increased, that is different thing. But if it is increased, acid is a must because that digests our food, that dissolves our food, that breaks down the food. So it is a must. We have acidic pH here. Okay. Now the thing is, when our body starts overproducing this acid, then this acid will give you acidity. And you will have a burning sensation in your heart. You will have a burning sensation in your stomach. If it is very high, in case if acid generation is too high, it can cause, a dissolution of this muscular layer. And just imagine If this muscles is dissolved, you will have open wounds inside your stomach, and that may even cause death. It was so severe. Okay. Overproduction of acid in the stomach causes irritation and pain to start with. In severe cases, ulcers are developed. This is very, very painful. Ulcers in stomach, okay, are developed. Until 1970, only treatment for acidity was administration of antacid, that we will just kill these acids. This acid is primarily HCl. Okay, so we used to give antacids like magnesium hydroxide. A pink color syrup under the name Gelusil, you must have all come across, or a pink big tablet. It is basically magnesium hydroxide. So we just want to neutralize this acid. So we will do MgCl2 plus H2O type reaction we are targeting. So until 70s, only treatment of for acidity was administration of antacids such as sodium hydrogen carbonate. Yes, how will sodium hydrogen carbonate function? HCl plus NaHCO3. So NaCl, strong acid displacing weak acid, plus H2CO3. But H2CO3 being unstable, water body will keep, CO2 you will burp out. Okay. So these were used. A mixture of hydrogen carbonate. Now, which antacids are common? Let me mark. Antacids such as sodium hydrogen carbonate or mixture of aluminum and magnesium hydroxides. Here it is magnesium or aluminum. However, excessive hydrogen carbonate can make the stomach alkaline because there was no measurement that how many equivalents of HCl is produced in the body. So it was difficult to provide how much should be given. If I give excessive of this, the stomach will become alkaline and then the digestion will be inhibited. Okay. And trigger the production of even more acid. If the pH of this medium becomes alkaline, the cells will think, okay, the pH should be acidic. They will produce more acid. Metal hydroxides are better alternatives because of being insoluble. They will not be dissolved under the aqueous medium. Okay, so it will be consumed only that much and will let the medium remain acidic. Metal hydroxides are better alternatives because of being insoluble. These do not increase the pH above neutrality. These treatments control only symptoms. Yes, that acid is being produced. They are killing the acid but are not removing the disease. That how to stop this overproduction of acid. Okay, and not the cause. Therefore, with these metal salts, the patients cannot be treated easily. In advanced stages, ulcers become life-threatening. Yes, as I told you, it can cause death. And its only treatment is removal of the affected part of the stomach. How to identify just affected part? If the process is like this, it can attack other parts also. A major breakthrough in the treatment of hyperacidity came through the discovery according to which a chemical, now here comes the king of chemicals, histamine, very, very important, stimulates the secretion of pepsin and hydrochloric acid in the stomach. So what is now clear कि there is a chemical जिसका नाम है histamine. This histamine signals stomach and produces signals stomach to produce what? So it's as the stimulates the secretion of pepsin and HCl in the stomach. The drug, a drug. So if I could attack on this histamine, I can control the production of HCl. That was the plan. That okay, let us attack the root cause. This is histamine. This is very, very important. Please remember this. And yesterday we were doing acidity basicity, right? So this is not basic. This is rather acidic center. It will release H plus. These two will be the basic centers of histamine. There was a question once long time back asked related to histamine's acidic basic centers in JEE Advanced. The importance of the drug was where we were, and the importance of the drug was so much that it remained the largest selling drug in the world until the another drug, ranitidine, was discovered. So first was cimetidine tegamet, okay, was discovered. Then the ranitidine was discovered. So basically what they are doing, they are somewhere acting as antihistamine. Antihistamine example. So original was cimetidine. 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 It was also Tegmet. Then came the ranitidine. Ranitidine. See this. If you see, these are big molecules. Both are having sulfur, CH2, CH2, NH, C, double bond, something. Now, this is CHNO2, this is N double bond C, single bond NHCH3. So these parts are common. If I can say this part is common, rather this is also common. Here there are some changes. In cimetidine, this was this histamine similar structure was there. Here it is something from furan. Okay. Antihistamines. Histamine is a potent vasodilator. Now, what is this term vasodilator? Vasodilator, what happens is, for example, our vessels, okay, vessels which carry our blood. Okay, these are veins and the arteries I'm talking. Over a period of time, if you talk of a simple pipe or something, in the sink of a home also, things deposit on their wall. And so is the case with humans, fats and lipids, deposit like this. Now, the passage through which the blood passes goes on thinning. Okay, and this can cause the heart attack. So these vasodilators, if are given to patient, then these all of a sudden expand and a bigger opening is created. Okay, this whole vessel is dilated. It has various function. It contracts the smooth muscles in the bronchi. Okay, vessels does not mean only blood vessels. It could be the air vessels in your lungs also, in the bronchi and gut, and relaxes other muscles such as those in the walls of fine blood vessels. Histamine is also responsible for the nasal congestion associated with common cold and allergic response to pollen. Histamine is also responsible for the nasal congestion associated with common cold and allergic response to pollen. Synthetic drugs such as brompheniramine, which is Dimetapp, and terfenadine, Seldan. These words, if you could remember, Seldan and Dimetapp, they are more common, act as antihistamines. So more to add here in antihistamine list is Dimetapp and Seldan. These are also antihistamine. This was Zantac. This was Tegamet. Okay. So this is how they function. Okay. This whole part is important, this para itself. Moving to next page. The next page is they interfere with the natural action of histamine by competing with histamine for binding sites of receptor. Okay, so they are basically competing for the binding site of histamine by competing with histamine for binding site of receptor where histamine exerts its effects. Now the question that arises is, why do above mentioned antihistamines do not affect the secretion of acid in stomach? Okay, we want that, okay, antihistamine if given, then the acid production should have stopped. The reason is that anti-allergic and antacid drugs work on different receptors. What an important question. Okay. If ultimately these are all functioning in a same way, that might be our thought, but no. If you are having something as an anti-allergic, it is also an antihistamine. And if you are having antacid, that is also antihistamine. But if you are having for allergy, it won't attack your acid or stomach pH because of this. Okay, no need to learn the structures, just as much as you can go in there. If the structure is given, how you will behave, that is more important. Few structures that could be important, we'll discuss. Neurological active drugs. So till now you have done mainly antacid or antihistamines, tranquilizers and analgesics. So tranquilizers and analgesics are neurological in it, they work on your brain. These affect the message transfer mechanism from nerve to receptor. Okay, tranquilizer are a class of chemical compound used for the treatment of stress, mild or even severe mental diseases. These relieve anxiety, stress, irritability, or excitement by inducing a sense of well-being. They will tranquilize you that all is well type feeling it will give. They form an essential component of sleeping pills. There are various type of tranquilizers. They function by different mechanisms. For example, noradrenaline is one of the neurotransmitter that affects a role in mood swing. If the level of noradrenaline is low for some reason, people say adrenaline rush, they feel excitement, they feel tranquilized to do something. Adrenaline rush. Noradrenaline is just opposite. If the level of noradrenaline is low for some reason, then the signal sending activity becomes low and the person suffers from depression. In such situations, antidepressants drugs are required. These drugs inhibit the enzyme which catalyzes the degradation of noradrenaline. In their body, if the adrenaline rush is less, they will feel depressed. So we send drugs which inhibit the enzyme, okay, which decompose noradrenaline in our body. If the enzyme is inhibited, this important neurotransmitter is slowly metabolized and can activate its receptor for a longer period of time, thus counteracting the effect of depression. What are the drugs? Iproniazid and phenalzine are two such drugs. Okay, iproniazid and phenalzine are basically these. See, they have a one thing in common, and that one thing is hydrazine type thing is there in both. Okay, some tranquilizers, namely chlordiazepoxide and meprobamate, are relatively mild tranquilizers suitable for relieving tension. Equanil is used in controlling depressions and hypertensions. If someone is having highly tensed lifestyle, they use Equanil. Okay, and these are mild tranquilizers, are relatively mild tranquilizers suitable for relieving tension. Equanil is used for controlling depression and hypertension. That is too much of tension if someone is taking. Right? Moving to next. So next point is derivatives of barbituric acid. Derivatives of barbituric acid, that is veronal, amital, nembutal, luminal, and second constant and seconal constant, an important class of tranquilizer. Now, first, what is this barbituric acid? So, what is this barbituric acid? Barbituric acid is obtained by reaction of diethyl malonate and urea. So what is diethyl malonate? CH2COET, COOET, and urea. What is urea? NH2CO, NH2CO. So when this NH2 attack and OET gone, here also NH2 attack, OET gone. So we have CH2CO, NH, CO, NH, CO. This is barbituric acid. Now here you can see that this site can act as acid. Of course, there are other sites also. But these lone pairs are in resonance. This, if I change, I will have different barbiturates. This is barbituric acid. When I make the alkyl derivatives of these, then they are called barbiturates. Now see, if you see this is the dialkyl derivative, diethyl. Okay, of see, if these two H are changed by 2 ethyl, then it is veronal. Very common. Okay, that is how it is made. Now, valium. If I talk of valium, it is different type. Hai na? Some other substances which are used are valium and serotonin. They are not barbiturates. Veronal is a barbiturate. Next is analgesic. Analgesics reduce or abolish pain without causing impairment of consciousness, mental confusion, incoordination, or paralysis of some other disturbances of nervous system. These are classified into two. So there are two type of painkillers. One is non-narcotic, one is narcotic. Non-narcotic, that even if you take, you won't become so habitual that you cannot live without it. Narcotic is like the negative extreme, that if you take it continuously, you will become so habituated to those drugs that now without them you cannot live. Then they are narcotic. So non-narcotic, non-addictive analgesic, aspirin and paracetamol belong to the class of non-narcotic analgesic. Aspirin, you have to remember the structure of aspirin. Aspirin is acetyl salicylic acid. This is aspirin. Acetyl salicylic acid is the most familiar example. Aspirin inhibits the synthesis of chemical known as prostaglandins, which stimulate inflammation in the tissue and cause pain. These drugs are effective in relieving skeletal pain, such as that due to arthritis, very severe pain. These drugs have many other effects, such as reducing fever. Fever reducing are called antipyretic. Pyre means fire, high temperature, and preventing platelet coagulation. Because of its anti and preventing platelet coagulation, because of its anti blood clotting action, aspirin find use in prevention of heart attacks. If you find someone around you who is just about to have attack, they are showing that I am having a chest pain or something, okay, give them aspirin immediately and then take to the hospital. That is the method of buying some minutes, some time, and the attack can be a little delayed. It is not that it will stop. That will be done only under medical supervision. But aspirin, 2 tablets of aspirin dissolved in a glass of water will give you some time to rush to hospital. Next comes a narcotic analgesic. Morphine and many of the homologs, when administered in medicinal doses, relieve pain and produce sleep. In poisonous doses, these produce stupor, coma, convulsions which are like somewhat like, what should I say, foot fits पड़ने जैसा, एकदम दौरे पड़ना, and untimely death. Morphine narcotics are sometimes referred to as opiates since they are obtained from the opium poppy. Opium poppy is basically a plant, and okay, and उसकी bud से हम इसे पाते हैं. Okay, what is opium poppy? I want to show you the picture. So these are like in Madhya Pradesh, we have a lot of such things. Close to Neemach, Neemach is a district where lot of these are grown. That is how the plant is. It is very, very commonly grown there. See? And it is under the government supervision, because for medical purposes, these are now very, very strong painkillers. These are very, very strong painkillers. Now, why I'm saying these are very strong painkillers? See, if someone is having a open heart surgery, if someone had a some skull surgery, so these are not the type of surgeries which can be, uh, handled with mild painkillers. नहीं हो सकता. Very, very intense pain is involved. So there they give these heavy painkillers. Of course, under the medical supervision. If medical supervision is not there, it should not be allowed. First, change your name and then I'll let you ask the question. Okay. So these analgesics are chiefly used for the relief of post-operative pain like cardiac pain and pains of terminal cancer and in the childbirth. Check the structure. If you check the, yeah, mommy here, please change your name and then I will let you ask the question. What is your real name? Kindly put your name. So if I see, here is morphine. If you see broadly, the structure is same in all the three. Heroin and codeine. All three are heavy painkillers. Okay. The only difference is here it has two OH. Here those two OH are now acetylated. And here they are, one is converted to ether. That is the only change. Otherwise, see three rings type structure. One is a furan, some types, some structure. So एक general picture you can have. I don't want you कि आप इसे याद कर लें structure को. No need to memorize the structures as such, but just a general comfort with the structures would be nice if you can have some. Okay, that would be nice for you to at least select out of the options. Now we are moving to antimicrobials. What are these antimicrobials? Let us start that. Diseases in human beings and animals may be caused by a variety of microorganisms such as bacteria, virus, fungi, and other pathogens. Okay? अलग अलग चीजें इस तरह की हमें डिसीज कॉज कर सकती हैं. एंड एंटीमाइक्रोबियल टेंड्स टू डिस्ट्रॉय or prevent development or inhibit the pathogenic action of microbes such as bacteria, antibacterial drugs, antifungal agents. These are what we will need. Antiviral agents or other parasites. Antiparasitic drugs selectively. Now, which disease is caused by what microorganism? On the basis of that, we decide whether we need antibacterial, antifungal, antiviral, or antiparasitic drugs. Okay, antibiotics, antiseptics, and disinfectants are antimicrobial drugs. Let us repeat: antibiotic, antiseptic, and disinfectants. So let us start with antibiotics. Antibiotics are used as drugs to treat infections because of their low toxicity for humans and animals. So they will not cause एकदम जहरीली नहीं है ना. They will not cause that level of infection or that level of activity on us that we ourselves get poisoned, but the small microbes will get poisoned. Initially, antibiotics were classified as chemical substance produced by microorganisms, bacteria, fungi, and molds that inhibit the growth or even destroy microorganisms. The development of synthetic method has helped in synthesizing some of the compounds that were originally discovered as products of microorganisms. Also, some purely synthetic compounds have antibacterial activity and therefore definition of antibiotic has been modified. Let us mark the latest definition. What is the latest definition? An antibiotic now refers to a substance produced wholly or partly by chemical synthesis, which in low concentration inhibits the growth, okay, or destroys microorganisms by intervening in their metabolic processes. Let us read again. An antibiotic now refers to a substance produced wholly or partly by chemical synthesis. Partly also because you may abstract them from some plant or some other microbe naturally available in nature, which in low concentration inhibits the growth or destroys, either inhibit the growth or destroy completely the microorganisms by intervening in their metabolic processes. The search for chemicals that would adversely affect invading bacteria but not the host began in the 19th century. The search for chemicals that would adversely affect invading bacteria but not the host, that it will attack the bacteria but not the host. This was started in 19th century. And who was the first one to do so? Paul Ehrlich, a German bacteriologist, conceived this idea. He investigated, very important, arsenic based structure, salvarsan aane wala hai, in order to produce less toxic substances for the treatment of syphilis. He developed the medicine arsphenoamine, known as salvarsan. Salvarsan was very, very important. Okay, syphilis was a major disease at that time. So, salvarsan used to treat syphilis. Okay, Paul Ehrlich got Nobel for Medicine in 1908 for this discovery. It was the first effective treatment discovered for syphilis. Although salvarsan is toxic to human beings, its effect on the bacteria, spirochete, which causes syphilis, is much greater than on the human beings. At the same time, Ehrlich was working on azo dyes also. He noted that there is a similarity in structure of salvarsan and azo dyes. See, salvarsan had arsenic in it. Okay, and azo dyes had nitrogen double bond nitrogen in them. So he was working on them. Azo dye here. I have N double bond N. Now what they did was these azo dyes and this arsenic, they had a structural similarity. He noticed that. Okay. Now after noticing this structural similarity, the arsenic double bond arsenic linkage represents present in arsphenoamine resembles the N double bond N linkage present in azo dyes, in the sense that arsenic atom is present in place of nitrogen. He also noted tissues getting colored by dyes selectively, like azo dyes. All tissues do not get colored, some selectively. Therefore, Ehrlich began to search for the compound which resemble in structure to azo dye and selectively bind to bacteria. In 1932, he succeeded in preparing the first effective antibacterial agent, Prontosil. Okay, first antibiotic is Prontosil, which resembles in structure to the compound salvarsan. Soon it was discovered that in the body, Prontosil is converted to a compound called sulfanilamide. In the body, it is converted to sulfanilamide, which is the real active compound. Thus, the sulfa drugs were discovered and a large range of sulfonamide analogs was synthesized. One of the most effective is sulfapyridine. Let us go back to the previous structure. Sulfapyridine. Okay, so basically it is an amide, sulfonic acid amide, right? Here, this sulfonic acid amide, and see, pyridine is there in it. Okay, that is how it was formed, right? Despite the success of sulfonamide, the real revolution in antibacterial therapy began with the discovery of Alexander Fleming in 1929 of the antibacterial properties of penicillin fungus. Isolation and purification of active compounds to accumulate sufficient material for clinical trials took 13 years. Okay, antibiotics have either cidal, that is, they will have a killing effect, or a static inhibitory effect, that either they will kill the entire bacteria colony or they will have some inhibitory effect, that that the bacteria will now be like less effective. Okay. A few examples of the two types of antibiotic are as follows: bactericidal and bacteriostatic. Static will stop, inhibit the bacteria, and cidal, they will kill the bacteria. Okay, so bactericidal is penicillin, aminoglycosides, ofloxacin. Okay, bacteriostatic, that they will inhibit, is erythromycin, tetracycline, chloramphenicol. The range of bacteria or other microorganisms that are affected by certain antibiotic is expressed at its spectrum of action. Antibiotics which kill or inhibit a wide range of gram positive, that is, एक dye होती है gram, if they get colored by that dye, then they are called gram positive. If they do not, then they are called gram negative. Okay, gram positive and gram negative bacteria are said to be broad spectrum antibiotics. So what are broad spectrum? Which kill or affect. Antibiotics which kill, inhibit a wide range of gram positive or gram negative bacteria are said to be broad spectrum antibiotic. Those effective mainly against only gram positive or only gram negative are called narrow spectrum antibiotics. Okay, those which are specific that they will affect only gram positive or only gram negative, then they are called narrow spectrum. If effective against a single organism or disease, they are called limited spectrum antibiotics. Okay, when only one particular bacteria. Penicillin G has a narrow spectrum, penicillin G. Ampicillin and amoxicillin are synthetic modifications of penicillins, and these are broad spectrum. So penicillin G is narrow spectrum. Okay, it is absolutely essential to test the patients for sensitivity to penicillin before it is administered. In India, penicillin is manufactured at Hindustan Antibiotics in Pimpri and in a private sector industry. That is not of much importance. Chloramphenicol. Here is a chloramphenicol. This is penicillin. And if you see penicillin, what functional groups are there? Amide is there. Again, an amide is there. Okay, sulfur is there. And carboxylic acid is there. Okay, here I see I have alcohol. All is there. Chlor. Okay, and there is chlorine here. Okay, chloramphenicol, alcohol, chlorine. Here also is amide and nitro. Okay, vancomycin and ofloxacin are the other important broad spectrum antibiotics. And the antibiotic desidezrine is supposed to be toxic towards certain strains of even cancer cells. It is even supposed to be toxic towards certain strains of cancer cells. Okay. We will do only till drugs today and we'll do the food and other part tomorrow. Okay, because I want you to repeat and revise again and again, then only it will be of any use. If I am doing continuously and you are not able to remember, it is of no use. It is already, you might be exhausted with the reading. Okay, so just wait for a few more minutes. We'll try to hold it once the drugs are done. Antiseptics and disinfectant. See, antibiotics, they were killing or inhibiting the bacteria. Antiseptics and disinfectant are also the chemicals which either kill or prevent the growth of microorganisms. Okay, antiseptics are applied to the living tissue. They are applied to the living stock, such as wounds, cuts, ulcer, like soframycin, and diseased skin surfaces. Examples are furacin, see, soframycin. These are all antiseptics. So they are not taken in. They are not ingested. They are just applied on the surface. Commonly used antiseptic is like Dettol. It is a mixture of chloroxylenol, this one, and terpeneol, like this one. Okay, it is a mixture of chloroxylenol and terpineol. Bithionol, the compound is also called bithionol, is added to soaps to impart antiseptic properties. This is added to soaps to impart antiseptic properties. See, these are the antiseptics used in Dettol. These together, these together will compose Dettol. And it is important for you to remember Dettol has chloroxylenol and terpineol. Okay, this is added in soap. This is added in soap. ठीक, is added to soaps to impart antiseptic properties. Iodine is a powerful antiseptic. Its 2 to 3% solution in alcohol mixture is known as tincture of iodine. 2 to 3% only. Okay, only 2 to 3 percentage is what we want. And here also I would say ki even it will come later, so just wait. 2 to 3% solution in alcohol water. Then it is called tincture of iodine. It is applied on wounds. Iodoform is also used as an antiseptic. Iodoform is also used as an antiseptic. Boric acid in dilute aqueous solution is weak antiseptic for eyes. Lens wash करने के लिए boric acid solutions are used. Disinfectants are applied to inanimate objects. Inanimate, I think, is a very simple word for non-living systems. Inanimate object, non-living things. Okay, so when it is said inanimate object, this इसका मीनिंग क्या है? नॉन लिविंग, such as floors, drainage system, instruments, etc. Like those sanitizers we use, those were disinfectants. Same substances can act as an antiseptic as well as disinfectant by varying the concentration. That is a possibility. For example, 0.2% solution of phenol is an antiseptic, while its 1% solution is a disinfectant. Okay, antiseptic apply on the skin, disinfectant on the floor and the furniture, all non-living things. Chlorine in the concentration of 0.2 to 0.4 ppm in aqueous solution and sulfur dioxide in very low are also disinfectants. Chlorine 0.2 to 0.4 ppm and sulfur dioxide in very low concentrations are disinfectants. Anti-fertility drugs. I think this is the last of the drugs and last for today. Antibiotic revolution has provided long and healthy life to people. The life expectancy has almost doubled. The increased population has caused many social problems in terms of food resources, environmental issues, employment, etc. To control these problems, population is required to be controlled. This has led to the concept of family planning. Anti-fertility drugs are of use in this direction. Birth control pills essentially contain a mixture of synthetic estrogen and progesterone derivatives. Okay, both of these compounds are hormones. It is known that progesterone suppresses ovulation. Synthetic progesterone derivative are more potent than progesterone. Norethindrone is an example of synthetic progesterone derivative most widely used as anti-fertility drug. The estrogen derivative, which is used in combination with progesterone derivative, is ethanyl lestradiol. Novestrol is the word that is easy to remember. See, novestrol has terminal alkyne, so it will give that tolerance test. They can ask the test, na, which one give what test? Terminal alkyne test. It will give neutral FeCl3 test. Both norethindrone and novestrol will give this neutral terminal alkyne test. Anna? They have a 3-degree alcohol, so you can have Lucas reagent test. But here it is a ketone and here it is a phenol. So there is a little change. Natural progesterone, okay, is structure is not given here of the natural progesterone. Here is a synthetic progesterone norethindrone, and then the estrogen derivative which is used in combination with progesterone is novestrol, and together they both act as anti-fertility drug. Okay, also read these in text questions. Sleeping pills are recommended by doctors to the patient suffering from sleeplessness, but it is not advisable to take its doses without consultation with the doctors. Again, because sleeping pills are neurological medicines and can cause several side effects. With reference to which classification has the statement ranitidine is an antacid been given? I think that we have read and you can give answer from there. Chemicals in food. These last 1, 2, 3, 4, 5, 6 pages I'll do tomorrow. We'll have a 1 hour class tomorrow and then we'll completely done. Okay, I think it's time for any doubt. If anyone has, please raise your hand. Please repeat your NCRT again and again, 2, 3 times. We need to read finally till your mains and advance. Anyone having any doubt? No one? Okay, Feast is saying something. Just wait, Feast. Let me unmute you. Yes. Tell.
Ma'am, in the structure of histamine,
Yes.
can you explain the site of acidic and basic nitrogen?
Yes, I can. Just wait. Let me go back to histamine. Here. See, this is histamine. You must be aware of what is your name, Feasty?
Ma'am, my name is Shubhangi.
Shubhangi. So, Shubhangi, you must be aware of this thing that pyrrole, this lone pair is part of aromatic cloud. So, this is
Yes,
not basic.
yes,
And
ma'am.
instead of base, it behaves as an acid. It releases this H plus easily as the anion is stabilized and is aromatic. Okay. So, coming to this NH, which is single bonded, is quite similar
It'll,
to that pyrrole.
yes.
Okay. And
The lone pair will take part in the resonance.
yes, yes. Excuse me. Now coming to this double bond, you were present in yesterday's class also, Shubhangi. So if you
Yes, ma'am.
remember, we did that guanidine thing now.
Yes, ma'am.
If from here it can donate,
Yes,
right?
ma'am. Okay. Yes.
And now wait one more thing. Now here the percentage S will be high, na? It is sp2.
Yes,
This
and this
is
is SP3.
sp3. So out of these two also, if they compare, na, which is a stronger. So please select this to be a stronger base. Then this. Oh, sorry.
Yes, even, but then the lone
Not
pair will be delocalized with the sp2 one.
But I'll withdraw that statement because I need to check doubt. I need to check the data. Reason is because this is equivalent RS. Na? It is like amidines are stronger base than primary amines.
Yes, we have that point yesterday.
Yes, yes. Yesterday we discussed. Na? So
Yes, ma'am.
I'll withdraw that statement and simply I'll say these two will be the basic centers.
Okay, ma'am.
Right, Shubhangi? Anyone else, better?
And ma'am, yesterday
Hmm.
the ketone enol form. So ma'am,
Yeah.
ketone forms more enol than aldehydes because I was reading
Uh, no, no,
some
no.
sandwich.
Ketones having more enol than aldehyde?
No, you said aldehyde has more enol form
Yes.
than ketone.
Ketone, yes.
So because I was reading my Singh's
Yeah.
reference book and it had the opposite of that written. That's why.
Oh. No, no. So please correct it because what we are using is from a very standard Jerry Marsh text. Okay.
Okay, ma'am.
So those seniors, which center you are from, beta?
I am from Bangalore Center. I'm in CV Raman.
Okay, beta. So just please correct it. Okay, because clearly we have very standard book. Maybe whosoever has taught might not have gone through this data and took you the thing that you were yesterday saying, that hyperconjugation thing.
Yes, ma'am.
Might be that is some people do that thing, but There is a condition in applying hyperconjugation that first they have to have the same functional group, then you can apply hyperconjugation, not before that.
Okay, ma'am.
Let us
Okay, that's all.
consider that was a blunder.